the wara were hunted to extinction by 1876, making them the first and only canid to go extinct in documented history. So for me, the situation of there being a fox on these isolated oceanic islands really raised the question, well, how and when did it get to the islands? And there are several theories regarding exactly this. And I'm not going to go into great detail on all of them, but they are worth mentioning. So the first is that it could have possibly swum the expansive channel from mainland South America to the Falklands. The second is that perhaps it floated that distance on a log or a piece of sea ice, or perhaps on a loose canoe. Then there's the idea that perhaps they walked across a land bridge or a sea ice bridge at times of lower sea level. And then there's the idea that most captivates me, which is the possibility that humans intentionally brought the wara from South America to the Falklands via some sort of sea craft. So if humans did indeed make their way to the Falklands, they likely would have done so via South America. And the first marine adaptations to show up in southern South America did so around 6,400 years ago. And these adaptations included the use of canoe technology, which, as you can imagine, is really important for my hypothesis here. The presence of the Falklands Current, which pushes around the tip of South America and at and around the Falklands, further suggests that it's not outside the realm of possibility that humans may have used canoes to intentionally or perhaps unintentionally venture to the Falklands. So the closest relative of the wara is a species called Symposium, which is another extinct species of canid from mainland South America. And the interesting thing is that it's commonly found in southern South American archaeological sites. And in one particular case, a Symposium individual was found as the primary subject of a burial, which basically means that it was found alone or buried alone, intact, and in a similar fashion to human burials at the same site, which was a mortuary site that humans have been coming back to for over 1,000 years. And burials where the animal is the primary subject are exceedingly rare throughout the archaeological record. So what this burial suggests is that this particular species of fox was capable of integrating into society and interacting with humans on a much more intimate level than simply that of a peripheral camp scavenger. So taking all of this into account, I hypothesize that humans dependent on marine resources may have intentionally, or perhaps unintentionally, followed the abundant wildlife that exploited the nutrient-rich waters making up the Falklands Current and found themselves in the Falkland Islands along with the wara or potentially Symposium. So I have three main questions that I'm seeking to answer through my research. And my first, I'll kind of walk through them incrementally one by one. My first question is there archaeological evidence of a pre-European human presence in the Falklands? And in a place where no archaeological evidence or sites have ever been found, and no archaeological studies, prehistoric archaeological studies have ever been conducted, this is a really difficult task, akin really only to finding a needle in a haystack, which I found out in gruesome detail on my most recent expedition to the Falklands. But before I went down there, so this was a trip that I went on in January and February, in order to pare down locations where humans may have actually chosen to live, I took into consideration things like proximity to freshwater sources, to hunting and fishing grounds, naturally harbored and wind-sheltered areas, gently sloping beaches, and places where artifacts had allegedly been found by landowners. I then visited these sites along with Dulcinea and performed a series of test pit surveys at some of the locations in order to determine if that site was archaeological. And as I sort of alluded to before, most of these test pits didn't yield anything of great interest. However, one site in particular was quite promising at New Island. Here we found 6 large bone piles, and we excavated, as you can see in this video, a 1 meter by 1 meter pit into one of them, and found the remains of numerous sea lions, fur seals, and penguins, which was really exciting. Now, these piles could potentially be from early European or American sealers, who would have then heaped the bones once they were done with them, and who were visiting the area in the 17 and 1800s. But a few things suggest that these piles could actually be a little bit older. First of all, sealers would tend to club or shoot sea lions and fur seals in the head. And none of the skulls that we uncovered, and we found about 15, none of them had any kind of skull trauma. Secondly, the piles had very large balsam bogs growing on top of them. And I apologize, this picture is a little bit dark, but it's that big lump. And balsam bogs are really slow-growing plants that grow up on top of themselves, and they can live upwards of 1,000 years old. So the fact that these bones are located under those bogs suggests that they could potentially be quite old, which is also exciting. And then furthermore, the bone piles are located in a place where 2 and possibly 3 stone points have been found on the surface by landowners. And that's one of the points found there. So I've collected several bone samples for radiocarbon dating. They're being shipped off to California, and only time will tell how old the piles are, but they have the potential to be very exciting. So in order to enhance the chances of finding evidence for pre-European human presence, I've turned to a different proxy, which brings me to my second question, which is there a distinguishable pre-European human presence evident in the charcoal record taken from peat cores? So at each target location, and a few other places as well throughout the islands, we collected peat cores, and in some cases had to dig gigantic holes when our core wouldn't work, and actually just excavate columns. And the reason that I'm looking or I'm interested in charcoal is that fire frequency tends to greatly increase when humans first arrive in a location. And this is particularly true for initial arrival to island environments. So I have this very rudimentary sketch of a graph here where you have fire frequency on the y-axis and time with the modern era on the x-axis. And you basically have really low natural fire frequency, and then at the time of initial human arrival, you see that spike in fire frequency. So I'm currently in the process of analyzing each core that we collected while in the Falklands for charcoal abundance in order to establish a natural baseline fire frequency. We can then use that baseline to determine if there are any unusual spikes in fire frequency that may indicate initial human arrival to the islands. So basically, to sort of tie it all back into the Wara, very few fossil Wara bones have ever been found. And so far, they've only been found at one location. So we've collected several samples and sent them off for radiocarbon dating, which brings me to my third question, which is, do the ages of fossil Wara bones overlap with evidence of a pre-European human presence? And prior to dating, we had no idea how long the Wara had actually been in the Falklands. But the dates that we have gotten back have helped to enlighten that situation. So we got 2 dates back, one from a pelvic bone and one from a shoulder blade that both date back to roughly 900 calibrated years BP. And then we got a much older date from a tooth, which dates back to 4,231 calibrated years BP. And this date in particular is quite exciting, as it's the oldest date that we have so far, and it basically tells us that the Wara was in the Falklands by at least 4,200 years ago. So at this point, that's sort of our best estimate for when the Wara actually arrived in the islands. So in conclusion, it's our hope that together the radiocarbon dates from the Wara bones, along with the archaeological analysis and the charcoal record produced from the peat cores we collected, will allow us to establish a chronology of human and Wara arrival times, and to determine whether or not the two may have potentially been connected. It will also help us begin to establish a natural paleoecological record for the Falklands that will hopefully contribute greatly to our understanding of the natural and human history of the islands. So before I wrap it up, I'm being told to clam up. Before I wrap it up, I just want to put a quick plug in for the Follow a Researcher program. Fellow grad student Charles Rada and I have helped the Cooperative Extension develop this outreach program that basically connects K-12 classrooms to graduate level research. You get to do all kinds of things, classroom visits, live Twitter sessions in the field with classrooms, and a whole bunch of other things. And we're currently looking for a researcher for this coming academic year to be the next researcher in the Follow Researcher program. So if you're interested in bolstering up your broader impacts for grants and stuff like that, it's an awesome opportunity to get to know some kids. So come and contact me if you are interested in that. So with that, thanks to everybody listed on this page, and a special thank you to Dan and Betty Churchill for making this research possible through the Churchill Exploration Grant. And I'll take any questions. Yeah.
Softball question, but it might be helpful for the non-archaeologists. There are no marks on the bones in that bone pile. Why do you think it's a human-caused pile?
So I think it's a human-caused pile, and this is something that, again, is a little bit up in the air, but mostly because of the bone assemblage and what was found in those specific piles. So we have heaps of bones that I can't think of a way that they would occur naturally. We also have the association of sea lion bones and fur seal bones, and the two populations don't typically intermingle like that. And so what we would need to do if the dates come back and they're interesting dates, we need to go back, look through for cut marks and marks on the bones, look for stone flakes or any evidence that it actually is human-made piles. So that's, again, kind of up in the air. But yeah.
Thank you. I think we probably need to move on.
All right.
Thank you.
All right, and next up we have Ben Partan. He's going to talk about Cathedral Glacier, a shrinking surface glacier in northern British Columbia.
Okay, I'm Ben Partan. I'm in the School of Earth and Climate Sciences. I'm looking at the Cathedral Glacier, which is a small glacier in northern British Columbia, over the time span of about 40 years from 1971 to 2015. So my objectives for this are to create a digital elevation model that can be used to determine how much mass has been lost or volume has been lost from the glacier. And I'll be doing that with time series. I have pictures over the last 40 years of the glacier that I can put together to make models from structure from motion, which is a technique I'll briefly cover later. And I'll compare that to how the mass loss is related to the temperature and precipitation changes over the years. So a brief outline of what I'm going to talk about. I'm going to talk about who cares about this glacier. It's so small. Where is the Cathedral Glacier? The temperature and precipitation data, the time series images, structure for motion, and then what do I do next? So why would we care about a glacier that's only like 1.2 square kilometers when Antarctica is 10 million times that size? Or even the ice field, the Juneau ice field, which is next to it, is 4,000 square kilometers. Small glaciers represent, there's not a good catalog of them, but there are estimates that they represent up to about 10% of the glaciers in the world. So if you're calculating your sea level rise and you forget about 10% of it, you're going to be off. And 10% is a decent amount to be off by. They also react faster to climate change because they're smaller. It's like stopping a car versus stopping a freight train. If you want to reverse the direction of your car, you just put on the brakes, you throw it in reverse. Where if you want to move a freight train in reverse, you got to really stop it, and it takes a long time. And that's the difference between a small glacier and, say, Antarctica. And currently, partly due to all the small glaciers, Alaska is the leading contributor to sea level rise at the moment. So where is the Cathedral Glacier? It's in northern British Columbia, right near the border of the Yukon. It's on the eastern edge of the Juneau ice field. It's in a continental climate, which means that in the winters are generally cold and dry, compared to the western side, which is a maritime climate, which is relatively warm and wet and gets a lot of precipitation. It's at about 2,000 meters, so it's relatively high. And it drains into Atlin Lake, which is the headwaters of the Yukon River. And let's see the next one. So looking at, this is from precipitation and temperature records, which are gridded, interpolated from historical ground stations nearby to create a continuous gridded record across the North America and the world. So this is 5-year moving averages of the temperature and precipitation. And I have the PDO, which is the Pacific Decadal Oscillator, which is a large-scale climate variability related to sea surface temperature in the sort of mid-latitude 20 degrees north to roughly this area around 60 or so in the Pacific Ocean. And when it's in a positive phase, it's typically where this glacier is, it's typically warmer and dry. And when it goes into a negative phase, it's cold and wet. And since 1976, it's been in a positive phase, so it's been warm and dry. And all these records are anomalies compared to a normal period, which I chose from 1921 to 1950. So they're how it's different from that period. And you can see that during this time, which is during the record that I have, the temperature has been above normal by about a degree or so, and the precipitation has been less than that normal. So you can imagine what that does to a glacier. And this graph shows what it does to the glacier. So the winter precipitation is, of course, the most important thing for accumulation on a glacier, because rainwater melts the glacier actually when it falls, so you need snow in order to build it up. And the snow level has been relatively constant at about 4 to 500 millimeters of water equivalent in the winter. And this is the level of melt, which is about 7,000 millimeters of water equivalent. So there's a big gap. That's the total melt. So the net melt would be about 6,000, 65. And even if that's way off by even triple, you're going to have a lot of loss. So this is how the glacier has reacted to it. You can see in 1971, it came all the way down here, turned this little corner, and then it starts to go back. And in 2015, these little black dots on the surface are in relatively the same place because the glacier has slowed its flow quite a bit due to mass loss. So this dot is the same as that dot, and you can see how much less there is down here than here, or up there, or somewhere around there. So these are pictures that were taken over the years that I can use to create structure for motion. Oh, structure for motion is the next slide. So I put the, this is the 1977. That was from a map that was created by the Junior Ice Field Research Program back then. And I separated it into zones so that I could do a model using temperature and precipitation and elevation with a lapse rate across it, because as you get higher, it gets colder. And I created 9 zones. And in 1977, say this one was at about 1925 in the middle of that zone. And today it's at about 1700, so 1750. So it's lost a little under 200 meters in elevation. Although these numbers might be slightly inaccurate, but the area change is fairly good. It was about 1.74 square kilometers in 1977, and it's now down to about 1.25. And we did a GPS survey this last summer to get better elevations. So, structure for motion. Structure for motion is a technique which you can make point clouds similar to what you'd make with a LiDAR laser system to create a 3D elevation model. And all you do is you take pictures, feed it into a software. The software looks for what are called tie points, which are points that are distinguishable in multiple images. And then it does all sorts of calculations as if you were holding a piece of paper and you change it. You know, you'll see perspective. So with that, it will find out how to make a model using all those calculations and how the things change due to perspective. And so I took about 1,500 images and I created a model of the cirque with the glacier in it, about 11 million points. And that can be used, you can stack them up with the previous models so you can see how they change from year to year. And so what's next? I'm going to make the intermediate models, which are slightly more difficult because the cameras and there aren't as many pictures and they're not from all the right angles, but I'll do my best. And then I'll calculate the rate loss in between the different 3D models so that you can get, say, from 1976 to 1979, it's lost this much, and then I can compare, and also got to determine some errors. But I can use the rate of loss calculated from the models to compare it to the mass loss using the temperature and precipitation data, compare them together, and then I can sadly maybe make a guess on when the thing's going to disappear completely. And last one. And I'd like to thank everyone here, especially the Juneau Icefield Research Program, which helps out with logistics and made this possible, and the Dan and Betty Churchill Exploration Fund, which funded my travel to Alaska. Questions?
All right, next up is Annie Boucher. Oh,
Giving Ben's talk.
yeah.
Go for it.
I bet I could get pretty close at this point. This is a picture of Ben doing his research. He's the one wearing the pink drone on the left.
Great. So, next up, Annie Boucher. I'm just going to talk about topographic fabric and bedrock weakness, distribution, and glacial erosion models.
Thank you, Lynn. My name's Annie. As Lynn said, I'm a second-year master's student working under Peter Coons in the Earth Science Department. And I'm going to be talking about glacial erosion models in Alaska. My project starts from 3 observations. And the first observation is that sediment discharge in the area, especially in the Copper River, is extraordinarily high. It's orders of magnitude higher than most other systems in the world. Ice flow patterns change through time. When we model this ice sheet in a glacier model, the velocity changes and the direction of flow changes. And the third observation is that the erosion models that we're using assume a homogeneous bedrock strength. And we know that that is a weak assumption. We know that there is localized weakening in the bedrock. And we just haven't figured out how to put it into the model in a way that's easy to do at this point. So my hypothesis coming off those observations is that modeling ice flow velocities at glacial maxima and minima can help us constrain the erosion patterns. And in fact, they are required to constrain the erosion patterns. And the second half of my hypothesis is that if we can figure out a way to approximate bedrock weakening patterns, that will also improve these glacial erosion models. So this is my field area, the Gulf of Alaska. Down here, the Aleutians kind of go off to the left, and the 3 major landmarks that I'm going to be discussing today are the Copper River. You can see the sediment plume coming out here, the Fairweather Fault region down here, and the Juneau Icefield all the way on the east side of the image. And this is kind of the erosion rate model that I'm basing this presentation around, and a lot of our modeling efforts are based around. And it starts at, so it's meant to calculate the erosion rate. And there's 3 things that go into it. The first is bedrock weakness, which is the K value. Then there's sliding velocity, which accounts for most of the forward motion of the glacier in this area, and the basal shear stress. And the basal shear stress we're not going to get into as much today, but I'm going to show you some images of glacial sliding velocities, and we're going to discuss the bedrock weakness. So first of all, the sliding velocity. We're in coastal Alaska, and we're going to zoom into this red box. And again, same landmarks, the Copper River, you can see the sediment plume a little bit better here, and the Fairweather region down here. And the important thing to see just right off the bat here is that the topography is generally trending kind of southeast, northwest. So we can look, the valleys largely run this way, the ridges run this way. That kind of makes sense. And I apologize first of all for the resolution of this image. This is glacial velocity. And you can see that a lot of the velocity is also following the topography here. So it's starting in the southeast and it's flowing northwest. And if we focus on this glacier right here, this is the glacier that follows the Chitina River through time. So this is the model stepping through time. Initially, it's continuing to flow parallel to the topography. But as the ice sheet grows, it starts to shut off in the middle here. It's turning blue
which is the slower velocity. And as we step forward, it ends up pretty much shutting off. And instead of draining to the northwest, it's now draining to the southwest, perpendicular to topography. And those changes in icefall velocity have implications for both where the sediment is coming from and where it ends up when it gets eroded. If we come back to our erosion rate model, the second parameter we're going to talk about is the bedrock weakness, and what changes bedrock weakness and where it's different, where it's weaker. And a lot of the weakness that we're looking at is fault dependent. So this is a tectonic map of coastal Alaska, and we've got the Pacific plate down here and the North American plate up here. And the Pacific plate is moving northwest at a terrifically high rate. It's converging at about 50 millimeters per year. And that causes a lot of fault damage, especially along the strike-slip system to the east. Excuse me. And that fault damage weakens the rock. And we can see this at the outcrop scale. I've spent 2 field seasons in Alaska looking at these outcrops, and we can see that fault damaged bedrock is weaker than intact bedrock, and it's weaker by about 2 orders of magnitude. So in practice, that looks like the fracture spacing kind of varying from 0 to 5 centimeters all the way up to more than a meter. And if we look at the cohesive strengths, the weak bedrock, it has a cohesive strength of about 10 to the 5 pascals. And the intact bedrock is 2 orders of magnitude stronger, so it's somewhere around 10 to the 7 pascals. But Alaska is a really big place, and it takes a long time to go to all these field sites. So we're looking for a way to scale up our field observations to a regional scale, something we could put into the model to compare to all those velocity measurements. So if we look at the fault map of Alaska, we chose 2 field sites. The first is near the Fairweather Fault, right in the middle of this fault system, and the second one is out on the Juneau ice field, kind of outside of the fault system. And those are meant to be compare and contrast field sites. And to get at this, because we can't go to all these field sites, we need to scale these measurements up. And so we use an elevation variance analysis. And the way the elevation variance analysis works is it starts at one point in the center, and it looks at the elevation variance between that point and all the points in a circle around it at a set radius. And it graphs the difference in elevation between that center point and all the outside points as an ellipse. And the ellipse is structured such that when the elevations are the same, the axis is longer, and when the elevations are different, the axis is shorter relative to the difference in elevation. So you get this ellipse at the end, and the long axis of the ellipse is oriented along similar elevations. And the ratio between the long axis and the short axis gives you the anisotropy magnitude. So how anisotropic is this topography? So you think that we would get a high anisotropy magnitude along a ridge and valley system and a low anisotropy magnitude in a plain. And so we can look at that anisotropy at every point on the map. And we can graph it as a bunch of ellipses, but that gets very complicated very quickly. So instead, we graph it as a color scale, a grayscale. So in this scale, white is high anisotropy magnitude, so it's a high ratio between the long axis and the short axis of the elevation variance, and the black is a low anisotropy magnitude. And we can see the Fairweather Fault region around here, and the Juneau ice field region down here. And if we pull out those two, so if we look at it with the faults, excuse me, you can see that the high anisotropy kind of lines up with the faults. We pull out those 2 images. You can see the Fairweather Fault zone on the left has more white spaces in the Juneau ice field, and Juneau ice field is in the middle of this image, and it's a darker, generally darker. So this is kind of lining up with our fault damage hypothesis, where we've got high magnitude anisotropy is more fault damage, and low magnitude anisotropy is less fault damage. So we expect more bedrock weakening in the Fairweather Fault and less bedrock weakening in the Juneau ice field. So if we pull back, we looked at bedrock weakness with the topographic anisotropy. We looked at the sliding velocity with the outputs from the ice sheet model. And we also can take phasal shear stress out of the ice sheet model. We don't have images for that today. But we get erosion rate if we calculate all these together. So at the end, we go back to our field sites. We can calculate the erosion rate at the last glacial maximum, this is one of our initial outputs from the erosion rate. And you can see that in the area, especially around the Fairweather Fault, where we had high ice velocity and we have high bedrock weakening, we get high erosion rates. And that pretty much makes sense with what we're expecting to see. You can also see in the Copper River, we're getting high erosion rates, which is where we're looking at those high ice velocities in the first set of slides. So in conclusion, we have a high resolution climate and ice sheet model, and that can significantly change our understanding of erosion rates and how erosion is distributed through time and space. Localized bedrock weakening because of fault damage both focuses ice flow and can locally increase erosion rates. And topographic anisotropy holds some hope for quantifying the critical parameters of bedrock weakening. And work remains to be done on quantifying exactly how bedrock weakness varies as a function of lithology and the anisotropy. Many thanks to all these people, but especially the Dan and Betty Churchill Fund. And any questions?
And I didn't understand what was motivating that model to change the course of the glacier.
So when the ice rides up, when the ice grows.
What is the model?
The model is the University of Maine ice sheet model.
Oh, okay.
It's the model that Jim Faster
Yeah.
wrote and that Sean is in charge of now.
Okay.
So as the ice sheet grows, the ice can override the topography, and which causes the change. I should have that.
Okay.
Obviously didn't. Does that make sense?
So that was a projection backward in time.
Yeah.
Okay.
I think you said that the difference in the erodibility was on the order of 2 orders of magnitude. Part of the zone. That assumes weakening of the same rock type, but presumably the rock types are much more complicated than that. What's the difference among rock types in the erodibility? It must be greater than that, or not?
There are a number of different lithologies. We are also looking at the anisotropy as a function of lithology. There are a number of different rock types in the area, but we think that the fault damage is at least as important, if not maybe more important than the lithology in terms of driving rock strength, controlling rock strength. Thank you.
I'm endeavoring, Dan, to stick to the amount of time.
Good.
All right, and next up we have Sky Heller on currents of the past, archaeological evidence for past changes in fish ecology in the Gulf of Maine, update and review of specialized protocol.
I'm actually not going to talk that much about the protocol because I started putting that together and thought, no, this is boring and they don't care. So I'm instead going to tell you about my last field season, which is ironically my last field season. Anyway, so my name is Sky Heller. I'm Brian Robinson's PhD student. You may know me because I've been here forever. I got a master's here and now I'm in my 3rd year of my PhD. And I am doing work looking at shellbins along the coast of the Gulf of Maine to try and understand past climate to help us plan for future climate. So the Gulf of Maine, prior to about 4,000 years ago, we find a lot of swordfish archaeologically. So here's an example of a swordfish sword eroding out of one of our shellbins. And this is really interesting because swordfish is actually a warm water species. And the Gulf today is a cold water body for the most part. So, although it's changing. So the hypothesis has been put forward partly by Dave Sanger, one of our emeritus faculty, that the reason that we had swordfish was there was a change in the currents in the North Atlantic, and instead of getting nice cold Labrador current water in the Gulf of Maine, we were actually getting Gulf Stream water, which is where swordfish travel up through the Gulf Stream, so that makes a lot of sense. So the hypothesis is that the Gulf of Maine used to be warm and then became cold at about 4,000 years ago rather abruptly because they disappear across the whole coast at about the same time. And since we're going possibly or probably towards a warm water Gulf of Maine in the future, makes this a really interesting period to try and study. Because of course the Gulf of Maine is an important resource for us here, especially for fisheries. So to do this, we wanted to look at what the archaeological record tells us about the ecology of the Gulf of Maine, partly for interest in archaeology, but for my purposes here today, mostly for interests in fisheries ecology. And we have records from excavations from a number of sites along the coast that have swordfish, but the problem is that these sites, if you can see, they were excavated in the 30s through the 70s, and most of the sites weren't screened. We usually screen our sediments through a quarter-inch screen in the field. So the only bone that they really recovered from these sites were cool-looking pieces that were big enough to see. So for example, the swordfish vertebra you see here. The kinds of bone that did not actually get recovered were tiny, tiny fish bones. And in fact, even today, when we screen through quarter-inch screen, the tiny fish bones don't make it back to the lab because they fall right through the screen. So what my work is doing is to bring the actual soil back to the lab. So take soil columns out of these archaeological sites, bring the soil back to the lab, and carefully screen them through very fine mesh geology screens in our lab, so that we can get things like, for example, this little alewife vertebra that is teeny, teeny tiny, and you wouldn't get unless you used these teeny tiny screens. So part of the reason we're doing this is to try and get at whether or not the fish assemblage that we see once we've identified these bones, which is hopefully going to start happening this summer, is going to be the kind of a cold water assemblage that we see today or a warm water assemblage that we're expecting to see. So for example, fish from maybe south of Cape Cod today. In the past, to try and understand if this was a warm water ecosystem. We were looking at what the species were like that we saw in this ecosystem. And this is important for fisheries planning for a couple of reasons. So first, we want to plan for sustainable fisheries. And no matter what the ecosystem looked like 4,000 years ago, it wasn't as impacted heavily by fisheries as it is today. So this can help us get an understanding of the baseline ecosystem of the Gulf of Maine before that baseline starts slipping with increasing fisheries pressures through time. The second aspect of this is that we want to plan for emerging fisheries. We know the Gulf of Maine is warming. We're seeing new fish species come into the Gulf of Maine. We recently had legislation on black sea bass, which is a new species coming north into the Gulf of Maine with warming waters. And if we can put these together, hopefully we can plan for sustainable fisheries, even with a changing future in the Gulf of Maine. So that's my hope. So we'll see. Stay tuned for updates. So a couple of problems with this is that we only have actually 5 sites. Oh, there we are. 5 sites along the Gulf of Maine coast, 4 in Maine and 1 down in New Hampshire, that have the types of samples that we need to do this type of research. So there are only 5 sites where small bone is preserved from this time period. Don't die on me, clock. There you go. And at these sites, we know that there's ongoing heavy erosion. So this is the waterside site that was dug in late 1930s, and here is the top of the bank during that period, and here's the top of the bank when we went back in 2013. So that's about 8 feet of erosion of that site over 70 years. And this is just going to increase with sea level rise and increasing storminess. So it's really important that if we want these samples, if we want to study this, we need to be doing it now. So we did the waterside site field work in 2013. The Turner Farm was dug in the 70s, but some soil was actually retained, and we have access to that soil. There's not a lot of it, but we're going to see what we can get out of it. And then this summer, we visited the Seabrook Marsh site, which is down here in New Hampshire, and also the Nevin site, which Chris told us about earlier. So I'll talk about the Seabrook Marsh site. So here you have the Seabrook Marsh site. Here is the lovely Seabrook Marsh. Here is the eroding bank at the front, and then behind this nice stand of trees is the Seabrook Nuclear Power Plant. NextEra Energy gave us permission to be on their property and dig with cameras, which we were pretty excited about because usually nuclear power plants don't want you taking a bunch of pictures on their property, and they have signs that say we will shoot you. So they let us on their property. So big thanks to them for trusting us to do this. And they're actually hopefully going to help protect the site too. And so we were able to go and dig. Let's see if this works. Yes. Okay. So we were able to go and dig at this site, and this is the site that actually has the adjusted protocol that I wrote about in my mini paper, because the problem with this site is all of the sediments have been basically underwater for 4,000 years. So most of the site is below the salt marsh. So where the salt marsh is eroding away, we can get at the site. So out here in the flat, where we have about 4 hours a day to dig during low tide, we actually are on the subsoil surface. So that's actually below where the actual occupation soil would have been. So the occupation soil was up in all of this, and it's washed away. But what we do find here are pit features and post molds that were dug down into the subsoil about 4,000 years ago when people were living here. And then we also there's a slumped portion, don't worry, salt marsh people, a slumped dying portion of the salt marsh that we took a column sample out of over here, which is really great because having column samples from 3 of the sites makes them very easily comparable, which is super important in science, as you all know. So we dug here during the 4 hours of low tide. We had to leave about 2 hours before 4 hours before low tide, and then we could dig during low tide, and then the tide would come back in, and that would be the end of the workday. That's it. You cannot dig anymore. And then we would sit up on the bank and wait and like eat our lunch and wait for it to come up high enough that we could canoe with all of our equipment back to the dock with our tons and tons of soil against the wind with tons of soil in a canoe. I learned a lot this summer, but it was a lot of fun to do. So. This is our Seabrook site. Hi, that was a lot of fun. I actually have come to adore canoeing. Okay. So here's some examples of what we found. So here is a pit feature down in the subsoil, and of course you can't see it because that would be too nice. So here is the dark pit feature. So this will have cultural material in it. And then we also found that in the eroding region, we would find artifacts. So here's our transit, and it's set up there for 4 days, and then all of a sudden we see, oh look, a nice plummet just on the beach where we've been walking every day. So we were finding cultural material there, but we also found in some of the samples from this feature some Chenopodia seeds, which are eventually became a domesticated species, but this is before it was domesticated. Blackberry seeds, and even a little tiny shark tooth, which I've never seen before in one of these sites, so that was pretty exciting. I'm not sure exactly what species yet, but it's teensy. So that was Seabrook Marsh. We also dug at the Nevin site, which I have 45 seconds to tell you about. So Chris already spoke about the Nevin site, but here you can see the front of the eroding bank. And then in this region, we cleared a little spot right at the very edge where there might have been maybe 50 centimeters that wasn't previously excavated in the front. We put in a tiny little excavation unit, isn't it cute? And got some column samples out of there. We were able to locate some of there's a really famous layer of pure fish scales at this site, and we actually found a piece of that layer, which is incredible. So we took a micromorph sample for that, which Andrea Heller is going to be analyzing, which is beyond my capabilities because I do bones, not soils. And so now the fieldwork is complete, and I have all of my samples, and my undergrads are going through them for me. Yes, I know, it's over, I have more. So my undergrads are processing these, getting the soil out, and then hopefully next year I'll actually have fish to tell you about. Any questions? Thank you to all the wonderful people and programs and associations that have made this possible. Yes, Sam?
Sky, that pit feature, what is the white bottom layer? Is that ash shell?
The white bottom layer. Oh my.
White.
Yeah.
I don't. A
You
glare
draw it
the
on there so
screen?
we can see the pit.
Yes, I will show you.
Is it?
It's not staying at her. Oh, don't point it at the screen. That doesn't help. Oh, there it is. Oh, this is a reflection because the soil is wet.
Ah.
I'm sorry. That's why I was like, wait, white bottom layer? Yeah. So all of this is sopping wet beach sand. So you have a black feature here you can barely see, and then this is dark red subsoil like that, and then this is just reflection from the soil being wet. Sorry, disappointing.
It's impressive how fast you can work and how much you can get done during one tidal cycle.
Yeah, you would not believe it. It's pretty intense. We have the pit to drain it, and then we build little dams up the pit and try and like scoop out the dirt, but eventually you just can't. It starts coming in the other side.
So that's the end of your work day.
Okay, thank you.
you.
All right, so next up, we have Jeff Martin. He's going to be talking about Project Bison, Bison Industry and Science Observation Network, a new stakeholder and citizen science collaboration.
Thank you, Lynn. Looks like I can figure this out correctly. All right, my name's Jeff Martin. I'm a second-year PhD student with Jacqueline Gill, both in the School of Biology and Ecology, CCI, and then EES. I wear a lot of hats, just like this one. And so what is Project Bison? In this instance, it's an acronym for Bison Industry and Science Observation Network, Bison. I'm concerned with what's behind the clothes, especially what's inside. In particular, the hock, the lower rear leg, there are 11 bones in that hock. It's known as your foot. It goes from the toes all the way up to the heel bone. They just happen to have most of their foot off the ground. The ballerinas of the prairie. So what is the stakeholder citizen science? It's a blended combination of stakeholder engagement, in this instance, bison ranchers, and they're helping design adaptive management strategies for the industry for what they can do at their farm. They're not concerned about the major economic drivers. And it's purely voluntary. I'm not forcing them to do anything. The citizen science portion, the bison ranchers are providing information, donating these hocks out of the goodwill of their heart, and information about the animal itself when it was alive and of their operation. The idea being that these citizens are going to participate in science about their industry and what they're doing. In particular, we're trying to combine both of our knowledge works. They see the animal every single day. I see animals from thousands of years ago. So this is my treasure, is their trash. The hocks don't have any meat cuts. They're typically discarded. If they are used, they're used for doggy bones. And I'm selfish, I don't want the dogs to be happy. They are indicative of body size. Every step that they take morphs the size and shape of that calcaneum and other bones in the hock. The bigger you are, the bigger bones you have. As well, in the fossil record, the bones are well preserved. These bones are super dense. They do not have hollow structures like the long bones do with the humerus and the femur. They preserve really well, and the whole structure does. So these are a one-to-one comparison through time. Looking at particular of the calcaneum, I'm not going to go through all the bones because that's way too much time. I do take a suite of measurements. They're very similar measurements that can be applied to every single bone in every dimension, ranging from as few as 7 measurements up to 30 measurements of the metatarsal. These allometric sizes are going to be what's controlling the body size, and body size is affected both by climate and vegetation. Bergman's rule, developed in 1847, says that in cooler climates, the trend seems to be that animals within that species, those members in the polar regions or in cooler climates, are larger. I wanted to see if this is holding true through the geologic time. So we remove space, we just look at climate for cooler temperatures. I presented this last year. For those of you that weren't here, we have 75,000 years ago with bison latifrons, the largest bison ever to live, horn span of 9 feet, going through the ice ages, going into the modern. The stable isotope here is delta oxygen, but it's flipped from what you're typically looking at it, showing that as it gets warmer, bison get smaller, and they follow these peaks and valleys fairly closely. So what I've been doing is developing a website that has all of this information ready to go for ranchers to go see on their own time. I'm also going to conferences, bison conferences, and being keynote speakers, giving other side presentations, letting people know about these geological events that are happening, that may be happening very, very soon with abrupt climate change going into the future. Why? Well, because I've seen that bison, or shown you that bison changed through geologic time with temperature change, How are they responding right now, and how will they in the future? So what I need from the bison ranchers is those hawks, and that is going to help establish a modern baseline of comparison. If I look at bison ranches from all over the U.S., I can start to tease out your regional differences, your geographic variance. And then from those regions, I can look at the climate and environmental ecology for what's driving their body size. This is the 67 sites that I've been using in the fossil record with over 1,200 specimens altogether. I'm trying to recreate that distribution with bison ranches. So far, I have 10 ranches that have agreed to participate and 4 associations that are also sponsoring this research as well in different modes. How does this work with the fossils? Well, I've kind of walked you through that. Using those 11 bones, the only thing is I don't have a ratio of mass to the bones. That's where I need the weight from the animals before it died and was processed for meat. I need to create that relationship so I can relate it to the fossils. So it's a one-to-one relationship. And then I can start to look at the spatio-temporal aspects of climate change. So what the ranchers are giving, 1 hawk from each animal that they process for meat, ship it to me, hopefully on ice. I don't want it to be stinky when I get it. I want a photograph of the animal before it was processed. I need to know how the animal was holding its weight. If the back leg is completely straight, that animal's overweight. They have to use their leg as a pillar rather than the typical Z shape that they have under normal conditions. I want the carcass weights, live weights, so I can create that mass ratio, the age of the animal, the weaning date. If they take them off the mother's milk, that's going to change the growth pattern of that individual, especially that early developmental part of their life. Number of calves produced if it's a female, male, females for sexual dimorphism, right and left. I can figure that out on my own, but I want to have validation. Their operation information, their contact information, and their address, that gets me to know exactly where they are. I can start to pull out from NOAA and other weather service stations, look at their climate trends that they've had over the past few decades. The associations that they go to, who do they communicate with? Where are their thoughts coming from on how their animals are responding? Getting that carrying capacity, so I want their herd size and their acreage for their operations. So far, the ranches that are reporting, I have between 26,000 acres down to 13 acres and 6,000 animals down to 13 animals. So it's already a big variability in what of the ranches that are participating. I also need to get at the number of bulls and cows that they have for breeding stock. I want to look at how restricted their genetic pool is going to be. The 13 animals, for instance, are probably having a fairly restrictive genetic pool to be diversified through. Need to know their primary and supplementary diets. If they are being grain finished, that's also going to change the meat of the animal, which is going to cause it overweight or underweight. These animals are going to be adapting to the environments around them and to the vegetation that they eat. And lastly, I need to know the auctions. I need to know what genetic exchange that they're having and what diversity that they could potentially have. What we'll be doing here at the University of Maine is processing those hocks to remove the soft tissues. I'll be doing most of it probably. Taking the measurements directly on those bones that I already indicated before, relating back to the animal biometrics. Analyze the morphometrics and climate through time or over the space of where they came from. Work with downscaled regional models. I'm still developing this and working on that. I saw Sean earlier and talked to you. Integrate biosphere climate data, regional predictions as we move forward with IPCC, different scenarios, and create those scenarios for which the ranchers, I want to develop the management strategies that I can assist them with different information to inform how they do this, which gets me to these. I've got, these are what I typically bring to my conferences for the speeches. It includes a quick little fact sheet. If they don't have internet, they can just mail me directly. I am snail mail friendly. I also have the stakeholder procedural packet, which outlines every single step that they need to do. So there's no mess ups along the way. Make sure all of the data is collected. And then a quick survey that is looking for those information about their operations. So the outcome is phase 2, which is probably several years from now. Establish and discuss the regional impacts using the regional associations as kind of the clearinghouse of information and meeting of the minds of sorts. I'm clearly in the data collection phase, so I don't have a lot of results for you other than those 10 ranches. And I have a lot of people to thank. It's been a not trivial undertaking so far, and will continue. Questions? Yes, Greg.
It seems like the human pressure, the human selective aspect of this would really complicate contemporary bison ranching, right? So how do you tease apart the rates of, say, diet versus breeding, selective breeding in terms of the outcome, both of which would have huge implications for size of the animal? How do you weight those differently in a follow-up analysis of this?
Yeah, that's one of the biggest things that I've been struggling with and how to do that. The photo that I'm wanting of the broad side of the animal for the conformation, how that animal holds itself skeletally, structurally, is what's going to get at that, I think, the best, even though it's not a true representation of the stressors from human selection. Humans, especially in the bison industry, are going to be tending to select for larger and larger and larger animals because they want the most meat per animal. I don't really have a solid answer for it. I'm still working on it. If you have thoughts, I'd be more than happy to hear that. Yeah, Matt.
You might have said this at the beginning, I might have missed it, but so what are you going to be providing to the farmers? What is the final product?
The final product.
Like, is there a way to, like, for example, to maximize their amount of the profit through, understanding how the climate's going to affect bison sizes? Or
Yep. So right now, what I'm looking like it's going to end up creating is a rather large report that I'm going to try and boil down to just mainly figures to get the point across as quick as possible. And economics are going to come into it. And if these animals are going to eventually evolutionarily become smaller, can we use that to the rancher's advantage? Instead of having 1 animal per acre, if we have smaller animals, can we now have 1.5 or 3 animals per acre, depending on what that difference give and take is going to be, and changing their management strategies for carrying capacity, I think is going to be the way forward, but the data still needs to come in.
has this been done with any other type of animal?
I'm sorry?
Has this been done with any other type of animal?
What I'm doing?
Broadly speaking.
Not at the level that I'm doing it, no. I'm not familiar with anything with cattle or sheep or goat.
Thank you. All right, and next we have Rachel Fowler and her presentation on elevated dust inputs to Southwest Greenland Lakes, effects on surface water, dissolved organic carbon concentration and quality.
Thank you, Lynn. All right, so I will jump right into it. So before I start this talk, I just want to lay a little groundwork about what is DOC, where does it come from, and why is it important for lakes. So dissolved organic carbon, DOC, can come from leaching of plant and soil organic matter into lakes. And it can also come from in-lake production, so made by phytoplankton, for example. And it's really important in lakes because it plays a huge role in carbon cycling. It also serves as a food source for different microbes, so it's very important for aquatic food webs. And as you can see from this gradient of DOC concentrations, it also plays a big role in affecting the depth that light can penetrate into the water column, which is important for things like thermal stratification and then primary productivity. So between 2001, 2003 field seasons and 2011 to 2014, this really large drop in DOC concentration in several Southwest Greenland study lakes was measured, 14 to 55% drop in these lakes. So keeping in mind why DOC is important in lakes, we are really interested in finding out what the mechanism is behind this drop. So while DOC was decreasing in the lakes, yokelups, which are a type of glacial outburst flood, started occurring in this area with a lot more frequency. So what happens after the flooding is that the water recedes back, and on the floodplain is left this really fine glacial till. And this can be entrained by the wind and blown across the landscape and into lakes as dust. So dust typically has a good amount of iron in it, and we know that iron can bind with certain portions of DOC, causing it to flocculate and precipitate out of the water. So we're wondering if this dust might play a role in the mechanism behind the decline in DOC that's been observed. And we also know that photodegradation can be really important for causing declines in DOC Arctic surface waters. Photodegradation has been shown to cause a 70 to 95% decrease in all carbon that's processed in some of these Arctic waters. So this led me to ask four main questions. How much dust is deposited on the landscape? How much iron is in this dust? And then how does dust deposition affect lake water DOC? And how might exposure to sunlight affect this process? So I picked 3 lakes as study sites. We have SS 1381 and 2 here, and SS 901 here closer to the ice. And 901 has a much lower starting DOC concentration than these other 2 lakes. And 1381 has a different DOC quality or composition than SS2. So my methods were kind of divided into 2 parts, a dust collection stage and then an experimental stage. So this dust collection got at the first 2 questions: how much dust is deposited, how much iron is in the dust. So I set up these dust samplers that face into the wind at near 901 and near SS2. And these catch airborne dust. So then for the experimental portion, I actually collected dust from the floodplain to use in the experiment. And this is what my experimental design looked like. And this gets at the 3rd and 4th questions that I asked. How does dust deposition affect lake water DOC, and how might sunlight be important? So the way I set this up is I collected surface water from the 3 study lakes that I just showed you. And I added that dust that I collected on the floodplain to the lake water. So that was my treatment. And then the controls were lake water with no dust added. And I set one set of treatment and controls outside, exposed to the sun, and another set I left in the lab in the dark. So I ran this experiment for a week, and I collected water samples from these bags on the first day as an initial sample, on day 3, and on day 7, last day of the experiment. So this is what I found for each lake. I'm just gonna kind of walk you through each lake and the patterns. So first I'll talk about DOC concentration, and then I'll talk about SUVA, which is a metric of DOC quality. And one way to think about SUVA is that a higher SUVA value is more of a terrestrial source for DOC, and a lower value is more of an in-lake source. And just to orient you with the x-axis, UC is a control exposed to sunlight. LC is a lab or dark control. UD is dust exposed to sunlight, a dust treatment. and LD is a dust treatment in the lab. So what we saw for SS2, which is here, was that DOC concentration increased in the sunlight exposed controls and dust treatment. And then for SUBA, this quality metric, we see a decrease. And this happened most in the sunlight exposed and the dark controls. So the controls are really the ones that decreased the most. And then for 1381, we see the same kind of pattern, whereas there's this increase in DOC concentration in the controls and dust treatments that were exposed to sunlight. But for the SUBA, we see kind of a different pattern than we did for SS2. So instead of both controls going down in the same way, the sunlight exposed controls and the sunlight exposed dust treatments both decreased. And we see a very similar pattern for 901 as we did in 1381. We have this increase in DOC and a very similar decrease in SUBA, where the sunlight exposed controls and dust treatments decreased the most. So just to give a couple more results and start to go into some conclusions, the dust samplers I showed you before collected between 3.9 and 5.6 milligrams of dust per day. The 5.6 was collected near the SS2 lake, and the 3.9 was from Lake 901, which was closer to the ice. The iron content in the dust is yet to be assessed. And then, kind of contrary to my hypothesis, dust did not cause the concentration of DOC to decrease in this experiment. Sunlight exposure also didn't cause DOC to decrease, but it might have important implications for DOC quality. So for this concentration increase, what I think might be happening is that we're seeing this bacterial DOC production that's greater than any photodegradation or bacterial degradation that's going on. And then for SUBA decrease, we have this difference that we're seeing between lakes. So in SS2, the sunlight exposure effect seems to be more important than dust addition, which flips for 1381 and 901, where dust addition seems to be more important than exposure to sunlight. And this makes sense, I think, because SS2 collected more dust than 901, so it's nearer to the road, it's in a dustier location. So adding more dust to SS2 might have an effect of lesser magnitude than to these other two lakes. So for this upcoming field season, I want to do another experiment where I have the same sort of setup, but I filter out bacteria to get a better handle of if bacterial production of DOC in sunlight exposed water is what's causing this increase in DOC concentration that I observed. So thank you, and I'll take any questions.
Nice talk. As a sedimentologist, I'm rather picky about the use of certain words. Till is sediment directly in contact with ice when it's deposited. The Okalaups are shedding outwash, silt, sand, clay. That's the term you should probably be using, outwash.
Great, thank you. Yeah.
Rachel, were you able to control for temperature in your experiments, and does that matter in these reactions?
Yeah, I think it does matter. I think it's important for the metabolism of bacteria. So the experiments that were left in the dark in the lab were at a pretty steady kind of room temperature. And then, I'll go back a couple slides. So these ones that were outside, I added water to these bins to kind of buffer the effect of temperature, but there still was, a daily up and down. So I do think that's important also.
Rachel, why do you focus on iron and not iron and aluminum?
That's a good question, because aluminum also is important.
Rachel, each of those tanks seemed to have 6 samples in it or something. I couldn't quite distinguish what was going on there.
So each of those bins, yeah, 6 samples fit into each one, but they were randomly assigned both the dust treatments and the controls. So
Were those 6 samples somehow separated from one another?
yes, they were in bags.
But were they all immersed in water,
Mhm.
in the same water body? Same water, or was the water separated?
You mean where I got the water that I put in the bags? Or they were in the water in the tub?
Yeah.
Yeah, yep, they were in the same water. That was just DI water taken from the lab.
Okay.
So,
Okay.
thank you.
All right, so next up we have Xavier Allegra. He actually just got to Maine a few days ago, and he's going to be doing an internship with Roger Hook. So he just arrived. His talk is entitled Arkansas River Outburst Flood Problems.
So I'm going to talk to you about the problem of outburst flood. So as an introduction to that, I can tell that ice age paleofloods are widespread problems. So for example, you can see on the background a picture that display layers of Missoula outburst flood and layers of Bonneville outburst flood. So most famous mega flood are the one that formed in the channel Scotland, so in Washington and Idaho states. So in that case of Missoula mega flood, the maximum depth was of 300 meters. So a neighbor, so to the flood that I will study is Lake Bonneville flood that happened near the state of Utah. You can see. Here you can see characteristic landscapes in the case of Lake Missoula mega flood. So on the foreground, you can see here, and here the post holes that are holes like carved in the ground. And in the background, you can see like a plunge pole, and the direction of the flow was like from north to south. So here is the structure of the talk. So there are 2 origins for boulders inside the flood. So there are iceberg rafting. So when iceberg carved inside the lakes, there were like boulders that were deposited inside the lake, and that boulder participated to the successive flood in the geologic history. So there was also that 3 glaciers flowing from the west and going inside the range at east were depositing moraines. Like there were like Twin Lakes moraine, Clear Creek moraines, and Pin Creek moraines. So this moraine participated as well to the flood that was overloaded. So here is how was reconstructed the elevation of the moraine from an empirical basis. So here is an example of an ice rafted boulder area. So the flow, the overflow was arriving from here inside this zone where we can find a lobe-shaped landform. So the size of the boulder we can see is a little bit decreasing downstream. So the farm to the west reflect the magnitude of the fluid and give a scale to the boulders. So here we can see that we can see here and here two ranges of mountain that furnished like the material for the fluid. So here is this landform here. It was carved out by the three glaciers that were arriving from west and arriving east on the east wall of the valley. So here is some location of splays that were overflowing over the terraces. So a mean age of boulders inside the splays was found to be more or less of like a mean age of 19.2 kiloyears. So that's it. So how can we model this fluid? Why should we do that? So modeling is firstly based on geographic empirical evidence. So we'll first use I will use open source USGS software Across. So the goal is to be able to better predict water downstream, and in order to prevent maybe societies from jeopardizing themselves. So for example, glacier lake outburst fluid are today a big risk in regions such as Himalayas or other places such as Switzerland and Norway. So here is an example of modeling of this fluid in the Altai mountain in the Shuya Valley. So here you can see that cross section here all along the valley were chosen depending on the size, on large enlargement or on the geometry of the valley. So we can see that there is a more closer cross section that are found, like when the geometry is changing fastly on the sides of the valley. And so here are some hydraulics behind the modeling. So we can see that Y is the water depth, Z is the bed elevation. There is a term involving V, that is the velocity head, and 1 and 2 are successive cross section, so beginning from downstream to upstream. So we can see that there is a modification of a quantity in that equation. Energy grad line is parallel to water surface, and we add like a term H. So here is an example of result with the Acrace software. So in that case, it's a steady flow calculation. So dashes are gravel bare surfaces, like all over here. So that is the data that was used. So the upper part are the red dashes, so lines that are maximum fluid level that were modeled on tops of iceberg. So that the arrows were used to trace this line. So the blue line is the present profile of Shuya River and the Kachin River. It's like the water depth surface. So and the green light is the terrace that was formed by deposition of bed load after flood maximum. So here are the references. If you have any questions.
All right, does anyone have any questions?
those boulders about 90,000 years ago? How were they dated?
I think like they date like with cosmogenic nuclide with like
Do you know which nuclide?
it's like decomposition of it dates like the exposure time of the moraine or of the boulder in the case of
10 beryllium.
sorry?
Is beryllium 10 dating?
Yeah.
Thank you very much. And next up, we have Megan Conway on niche evolution along a gradient of ecological specialization.
What determines a species range is one of the fundamental questions in ecology and is gaining increasing interest with the threats of climate change. Species range is broadly defined by its ecological niche, which is a set of biotic and abiotic factors which a species needs in order to persist. An ecological specialization is often synonymous with a narrow niche breadth. So it's also often thought to occur a trade-off where a specialist gains a competitive advantage to take advantage of an abundant food resource, but might, by specializing on a narrow set of resources, might lose the ability to use alternate resources. So over the long term, this can come at a cost where a species might have a reduced ability to adapt to environmental change. For example, if you're a sparrow that nests on the ground in a tidal marsh that gets inundated by sea level rise, you might have a little difficulty raising offspring and reproducing. So as a consequence, ecological specialization has often been associated with increased extinction risk across a variety of taxa, including in tidal marsh taxa. This is a study by Mo Carell, where species on the left that occur over a broad range of habitats are showing fairly stable or increasing population trends over time. And those on the right that are restricted to tidal marsh habitats are showing fairly steep declines across their range. But we do know a species niche can change over both space and time. So determining what factors constrain a species niche is important both to predicting how species might respond to environmental change in the future and which species might be most vulnerable to those changes. So the central question here is niche width correlated across different axes? So is a specialist always a specialist no matter what trait you pick, or is a species only constrained by maybe its narrowest niche axis? So it's often assumed that a generalist, like the song sparrow on the right there, has a broad environmental tolerance to any trait. So if you're talking about maybe temperature or precipitation, the broad tolerance across both of those traits might allow it to inhabit a broad range of ecological conditions. The opposite might be true for a seaside sparrow or specialist that might have a narrow environmental tolerance to a lot of different niche axes, and that might restrict its range to a small area. But very few studies consider the middle scenario where a species might show broad environmental tolerance to one trait or environmental condition, but show a narrow tolerance to another. So this is could be a big criticism of niche modeling, where ecologists often try to predict how species might respond to change. Because you can imagine across all of these different scenarios that depending on the direction and the magnitude of change, that each of these species might respond very differently. So for example, you have a red species and a blue species with different niches, and a yellow circle that represents the environmental conditions in which those species exist. If you have a scenario where the environmental change occurs along an axis where species might have a broad variation or environmental tolerance, that species might have species one might have a high probability of persisting following that environmental change. Whereas if that change occurs across an axis where that species has very narrow tolerance, it might have a much lower probability of persisting following that change. So by comparing across species that vary in their ecological specialization, we might be able to predict how species might change through, or how species might respond to environmental change, and are specialists indeed, do they indeed lose the capacity to adapt over time the longer they specialize on a specific habitat? So tidal marshes are an ideal system to explore these questions because they're ephemeral over a geological timescale. Some of the northeastern marshes were covered in ice up to about 15,000 years ago. They're highly productive ecosystems, but the harsh environment provides a barrier to colonization. So there's very low species diversity and very few that have figured out how to adapt to life in tidal marshes. Also possesses several ecological gradients that can shape a species niche. One example is salinity, and tidal influence, which decrease as you move upriver from the coast. And there's also been several independent colonizations by sparrows that are closely related. These provide a gradient of ecological specialization that can reflect how that ecological gradient might shape a species niche. So you have the seaside sparrow that colonized about 2 million years ago, up to the song and savanna sparrows who are more recent colonists. The ones on the left are kind of restrictive to very coastal habitats. And as you move right, more of those species use upriver, brackish, tidal marshes, and a broad range of other habitats outside of tidal marshes. So we can quantify niche across multiple axes. We identified a suite of traits in the literature that are important in adapting to life in tidal marshes. And we can measure measure a species niche directly by the proportion of habitat that it inhabits, as well as indirectly through a suite of both morphological and physiological traits that might reflect a species' ability to adapt or live across that environmental gradient. So we might predict that if niche width is correlated, generalists that show, so this is each of these lines is a pairwise comparison across one niche axis and a second niche axis. This is a line across species, so each dot is a species, and you would expect the species that are up here will have a broad tolerance on one axis, would also have a broad tolerance on the second axis. Your specialists would be in this corner where they have a narrow niche breadth across several different environmental traits. But when we look at our data, again, these are pairwise comparisons across a multitude of niche axes. It really depends on which traits or which environmental gradient you're looking at. So we do see that some generalists are generalist across a couple of traits, but it's not always true. So we can break this down and look at specific traits and see how they vary within a species across that gradient. So this is a measure of hematocrit that reflects a species' ability to osmoregulate. So that's important in a high salinity environment where there might be limited fresh water available. This reflects the salinity gradient measured by vegetation characteristics. And you can see two of the species, seaside sparrow and salt marsh sparrow, on the top that are restricted to tidal marshes. They were the earlier colonists. They've been in the tidal marshes longer. They're highly specialized, show very little variation in hematocrit and very little sensitivity to dehydrating in very saline environments. Where more recent colonists like the Nelson sparrow, who inhabits more brackish river marshes, starts to show a little signal of dehydration once the salinity in an environment increases. So in conclusion, we do see some evidence that specialists may have narrow niche breadth across multitude of axes, but defining what a specialist is might depend on the traits and environmental variables that you choose. And so looking at variation within species across a gradient might provide more insights into what constrains a species niche. So with that, I'd like to thank my field crew, collaborators, and my lab mates.
Yeah, how can you know that a species has been specialist for a million years?
Genetic data times the time of divergence, estimates the time of divergence. Anyone else?
All right, next we have Dulcinea Groff on how do land use and seabirds explain the chemical characteristics of soil and use of grasslands in the Falkland Islands.
Right, so I'm sticking with the theme of tussock grass, but this time I'm taking more of a modern approach and addressing a topic that is of use to Falkland Islanders. Whoops. So, this is Ben Bernston. He's a landowner who is invested in cattle ranching as well as ecotourism. So you can see here in this image, we have cattle in the background and sea lions hanging out together in the wintertime. So this work is in the Falkland Islands. The Falklands, one of the top 3 sources of revenue comes from cattle ranching and sheep farming. And this has had a really great impact, a horrible impact on the tussock grasslands. And these right here are these little clumps that you see are the tussock grasslands. And they used to occur along every island. But today they're only found in like these small populations on the 2 main islands. And then on offshore islands, they're quite abundant where grazing hasn't occurred over the last 140 years. So tussock grass has a really important purpose for humans in the Falklands. And the first one is it stabilizes land in a region where the winds are extremely fierce. And you can see in this image here of Volunteer Point, this is a beach that over recent years has eroded further and further back. And this is essentially property loss. Another ecosystem service of tussock grass is that it provides habitat for wildlife. So when an individual landowner is not maintaining their cattle ranch or their cattle or their sheep, in the summertime they're entertaining and guiding tourists around looking at the wildlife that occurs on their land, such as the Magellanic penguin, which burrows in the tussock grass, and other seabirds that are important. And the last ecosystem service is fodder. There are hundreds, thousands of sheep in the Falklands and also cattle. And over the last 140 years, like I said, these tussock grasslands have been decimated. And anecdotally, tussock grass is one of the most nutritious plants in the Falkland Islands. So I've mentioned that historical grazing is a threat to tussock grass, but also declines in seabird populations could also pose a threat. So for example, the southern rockhopper is a species of penguin where in the Falklands, 40% of the global population occurs here. And in over recent years, these populations have declined. It may be an outcome of shifting prey items or declines in prey items. And so this means that there are fewer nutrients coming into the landscape from the biologically productive waters of the Patagonian shelf. Climate change also poses a threat to tussock grasslands through increased disturbances. This allows or provides a platform for invasive plants and pathogens to come in and take over. For example, there's a fungal rust that occurs across tussock grasslands, and it has the potential to become to spread even more in the future. And on top of that, the number of introduced taxa has more than doubled in the last 40 or 50 years. So the farmers in the Falkland Islands came together to discuss their concerns that they have over climate change and how that will affect their farming, their livelihood. So with a 2-degree increase in temperature, the Falklands are projected to change from a dry tundra to a cool steppe. And this poses a threat to increasing the soil aridity and erosion, as well as changes in the plant communities. So you could have non-native plants coming in or native plants shifting their ranges. Also, agricultural intensification is a threat, and habitat disturbance from increased storminess or increased fires. Through a vulnerability assessment of native plants in the Falklands, tussock grass falls into the category of moderately vulnerable. And as I've mentioned, this has some potential impacts on the ecosystem services that people benefit from. And there are other taxa that are highly vulnerable, but they may not play as an important of a role as tussock grass does in an ecosystem. So in the past few years, people have been trying to restore tussock grasslands, and some efforts have not been very successful, and some efforts, though, have the potential to be more successful than just replanting. So one land use type or one method of increasing tussock grass is to use rotational grazing, where only in the winter time, between May and August, for several weeks, probably, I think it's about 8 to 10 weeks, ranchers will allow their cattle to enter these fenced-off tussock grasslands to eat when other fodder is less available on the landscape. And so then there are also other tussock grasslands that have never been grazed and that are really what I'm considering pristine. And these grasses here are really, really tall. You could probably barely see me if I was standing here in this image, just for scale. So I set up a study design to look at how things like land use or the presence or absence of seabirds might influence some of the soil and plant characteristics in these different types of land uses. And we did this across 5 different sites in February, very recently. And what we did is we measured out 5 meter by 5 meter plots and measured the seabird nesting density, which is just the number of burrows within a plot, and the tussock grass cover, the percent cover of every species that was in that plot. And we randomly selected our plots. This is an outline of tussock grassland. And we randomly selected the plots. And this is just an example of a rotational grazing site. Here's a house. And this site was actually, it was planted 100 years ago, and it's been rotationally grazed for about a decade. Within each of those sites, we also collected soil and plants. And with each of those samples, we're going to look at like total nitrogen, phosphorus, and potassium, as well as pH. And I also extracted exchangeable nitrogens like ammonium and nitrate to have those analyzed in the future. I was fortunate enough, I don't know, it's not clicking. Oh, there it goes. I was fortunate enough to use the facilities at the Department of Agriculture in the Falklands to do these extractions during our last week of our expedition. So I just have some preliminary analyses to show you just basic plant cover in 3 of these land uses. And I'm not very comfortable with showing my other data yet, so I need to sit on it for a little bit longer before I explain it. But I'll go through this graph with you. I think this one's really interesting. This is an ungrazed site, and green is a tussock grass, and tussock grass is a native species here. And there's a less percent cover of invasive plants in the ungrazed places than in the historically grazed sites or in the rotational sites. So if you're thinking about biodiversity, it's really interesting to conserve these tussock grasslands because they really dominate a site. They really dominate the landscape where they shade out other plants and not many other things can tolerate.
What is the x-axis on those?
The x-axis, these are just the individual plots within a site. So we measured between 12 and 20 per site, 12 or 20 plots. And so my future analyses, I'll be looking at the responses to land use and seabirds, such as plant cover, bare ground, tussock cover, and native and invasive species, as well as these other things that I mentioned earlier. And my goal is to hopefully address some of the landowner concerns about invasive plants and soil erosion, and maybe shed some light on whether rotational grazing is actually a feasible compromise to maintaining a livelihood of having livestock on a landscape that has not supported herbivores in the past, and whether the presence or absence of seabirds plays an important role in that as well. And I'd like to thank my advisor, Dr. Jacqueline Gill, my committee, Catherine Hamley, for enduring some great field work, and Corianne for her advice on extracting nutrients, and lots of people in the Falkland Islands, especially landowners.
In New Zealand, there's a lot of sensitivity to tussock, to oversowing, superphosphate oversowing. So do they do that?
Super what?
It's a fertilizer with a superphosphate is referred to, but it's a commercial fertilizer, and the tussock ground is, for a number of reasons, is very sensitive to that.
As in, so like in some places where seabirds occur, there's too much nitrogen or phosphorus, and it basically just devastates all of the vegetation. Is that what they
That's
found?
related, yeah.
Yeah.
It's associated with the overgrazing as well.
Mhm. Yeah, it'll be interesting to see if different types of seabirds plays an important role in this, like whether it's a burrowing seabird or like a colonizing seabird. We could have very different impacts on tussock grasslands.
So those soil samples, are they organic soils or mineral soils? Percent carbon.
I think they are organic soils, and actually a lot of them are just peat.
Yeah.
So haven't decomposed very much.
Sure.
Mhm.
Thank you very much. All right, so next we have Elena on a 2060-year record of atmospheric arsenic deposition from South Pole Iceland.
Hello, my name is Elena Korotkik, and today I will be talking about arsenic.
Yes. Let me see. I restarted. Oops.
So arsenic is one of the most toxic elements in the environment and could be found everywhere in geosphere in a small concentration. It could enter environments through both natural sources and anthropogenic sources.
Natural sources include wild forest fires, soil dust, sea spray, but the most important sources is volcanic emission and biogenic emission, both continental and marine. The most important source for anthropogenic emission is a non-ferrous metal production, especially copper production. The second most important source is a fossil fuel combustion, especially coal burning. And recently, several studies have been reported elevated levels of arsenic in the air, groundwater, and soils, and in most cases, it was attributed to anthropogenic activity. Maybe. So in the late 20th century, it was estimated that 40% of total arsenic emission comes from anthropogenic sources and 60% from natural sources. And arsenic has a residence time about 10 days, so it allows it to travel over a long distance. And ice cores is a good opportunity to measure arsenic concentration in the remote areas. So here I will present South Pole arsenic record. The South Pole, 200 meters of the South Pole core was sampled at a high resolution, and samples were analyzed for major and trace element content and major ions. The core was dated in usual seasonal variation in the sodium, chloride, and strontium, and we also use major volcanic events as the age markers. So this plot shows the South Pole arsenic concentration. The thin gray line represents raw data, and the green line represents background level estimated using robust light smoothing function. As you can see, the arsenic level at South Pole are very low, and the mean concentration for the whole period is 1.7 nanogram per liter. And concentration are the same magnitude with other arsenic concentration reported from other Antarctic sites. But other Antarctic sites only cover a much shorter period of time, so South Pole is the longest record so far. And so the major question is, what caused this arsenic variability? Is it related to natural processes, or it could be anthropogenic? And to answer this first, because one of the major source for arsenic is a volcanic emission, we calculated volcanic input. So arsenic could come from both major volcanic eruptions and possibly degassing volcanoes. So first we compare changes in arsenic concentration. You can see the green line with the during the major volcanic events. You can see the non-CSO sulfate red line. The major peaks represent the volcanic events. And as you can see, during the 19th and 20th century, there is no arsenic increase during the volcanic eruption. It could be explained that during this time, anthropogenic emission was quite large, so it masks the natural signal. And in the earlier volcanic events, we do see increase in arsenic corresponding to volcanic eruption. So our data suggests that major volcanic eruptions could cause short-time increase in arsenic concentration. Next, we wanted to estimate contribution from degassing volcanoes. And it's very tricky because, so to do this, we compare arsenic to sulfur ratios in volcanic plumes to arsenic to sulfur ratio at the South Pole. But as you can see, we use all available data, and all the records, they show quite a wide range of the arsenic to sulfur ratios. So it's really hard to do the estimation. The highest values were reported from Mount Erebus, which is located in Antarctica. So we use this, we use the arsenic to sulfur values from Mount Erebus to estimate our volcanic contribution. We also estimate sea salt contribution using sodium as a reference element, and contribution from salt dust using cerium as a dust reference element. And according to our calculation, sea salt contribution to South Pole arsenic is negligible, it's less than 1%. Salt dust could contribute up to 4%, so it's quite small. Volcanic contribution could be very important. It's in general about 30%, but it's especially during the period of low arsenic deposition, volcanic emission could contribute almost up to 100%. And our data is very similar to estimation made before. You can see in this table. Unfortunately, it's not possible to estimate biogenic contribution, but it could be very important. So here we calculate excess arsenic. It's after we remove sea salt, dust, and volcanic part from the total arsenic. So that's what we have left. And the question is it all biogenic emission and forest fires, or could it be some anthropogenic activity? And the most interesting feature in South Pole record is this large increase since 1970s. It's highlighted in red. So in this time, the arsenic concentration really dramatically increased. And this increase also shows in the other Antarctic records. You can see on this blue color plot, there's a Don Fuji record from Japanese team, a couple ITAS record. So it's not just local, it's a global increase. And because the major source for arsenic is the copper production, and Chile is a major copper producing country in the world. So we compare Chilean copper smelting production with our arsenic, and we have very good correlations. So you can see the same trend, it's slowly rising and dramatic rise since 1970s. And the second important source is the coal combustion. We compare this South Pole arsenic with coal combustion in South Africa and Australia, and we also see very good correlation. So we can say with we're pretty much 100% sure this increase from 1970s, it's 100% anthropogenic and it's caused by copper smelting and coal combustion. We also see increase in lead, and copper at the same time, further confirming that it is anthropogenic. But the question was before 1970s, is it all natural or not? To answer this, we compare arsenic with lead, manganese, cadmium, zinc, and copper. It's elements which could be influenced by human activities. And we see elevated values to the copper and lead from like early 19th century, which correspond to higher values with arsenic. And we know that in the 19th century, in the mid-19th century, there was a boom in mining in Australia, which could cause some of these elevated values. Also, from like 1820s to 1850s, they were introducing new type of furnaces in Chile for copper smelting, and it required a lot of wood. So they, for a period of 30 years, they cut a lot of wood, and then also in the central Chile. And that's exactly where we see the big peak with lead and arsenic concentration. Another interesting part is we see very significant increase in copper, zinc, cadmium, and manganese from like late 13th century to up to about 1450. And this exactly the time when we see higher arsenic concentration. And this time corresponds to initial burning period in New Zealand. It was after Maori arrived to New Zealand about 750 years. Apparently they initiated a lot of forest fires. And for the short period, for about 180 years, they were burning a lot of wood, a lot of forest, and they managed to destroy up to 50% of the forest. And so it's a major event, and it's exactly when we see this large increase in a lot of elements. Before this, we do see some increased arsenic concentration, but not in the other elements. And so we suggest it's probably related to natural, so it's probably caused by natural sources. We do see some small increase in lead, but but also in sodium, which is sea salt input to South Pole, and cerium, which is indicator of dust input. So it suggests that this arsenic increase probably caused by intensification of atmospheric circulation. So more arsenic from natural sources will arrive at the South Pole. And most likely the same was about 300 years. So in summary, the arsenic story is quite complicated. Our data suggests that that biogenic emission and volcanic emission are major sources for arsenic in Antarctica, but anthropogenic activity could also contribute significantly to arsenic budget, and not only in the late 20th century, but in much earlier period of time. Yeah, thank you.
We have time for questions if anyone. All right. Thank you very much. We're going to deal with the technical.
Okay. I'm going to have some animations, so we'll have to work as a team. Should be good. Yeah.
All right, so we've got a couple of technical difficulties, but we have one more talk. Benjamin Seliger is going to take us to the end with how effective are United States protected areas at conserving nature's environmental stage?
Hello, everybody. My name is Ben Seliger, and I've been a PhD student with Jacqueline Gill here, and I've been doing research on tree migration and whether or not trees are going to be able to keep up with climate change. And today I'm here to talk about some new ideas that I've been having. And so that's why the title of my talk is a question. I hope to convince you that this is an important question worth asking in my talk. So next slide, please. And so I'm going to start off with a species that we're probably all familiar with, the Bengal tiger. And the Bengal tiger is highly endangered. There's actually more individuals in captivity now than there are in the wild. And one of the major threats to Bengal tigers are poaching for their skins and also killing them just because the people around there don't like living with a predator that views them as food. And so if you were put in charge with the task of conserving the Bengal tiger, your first thought probably would be to cut back on the poaching and hunting pressures, right? Well, I'm here to say to best conserve the tiger, next slide, you should probably think about conserving some of the species that it eats, like some of these tasty deers here. And some of these deers are vulnerable themselves and near being endangered. And so I'd say if you want to help bring back these deer, next slide, you should probably help bring back the Sundari trees, which are mangrove trees in the area, which provide kind of the structure for the ecosystem and also provide a lot of fruit. These trees, this is all water down here. And so without these trees, there's not much structure to the ecosystem and it floods a lot. And next. So in order to best conserve these trees, I would argue that you have to conserve this entire Sundarban mangrove forest ecosystem. And this is the largest intact saltwater mangrove forest in the world. Next slide. And so what I've kind of set up here is a dichotomy from fine filter conservation to coarse filter conservation. Fine filter conservation, you're focused on the needs of individual species. Coarse filter conservation, you're focused on entire ecosystems. And this is actually kind of an idea that was first kind of proposed by our very own Matt Hunter in the Wildlife Department and George Jacobson, Professor Emeritus at CCI. And next slide. When you conserve the entire ecosystem, you also help conserve other endangered species within that ecosystem. So the idea is that you're coarsely saving the entire ecosystem. And I would argue that without a coarse filter conservation approach, you probably wouldn't be able to conserve these species because our fine filter conservation approaches are oftentimes targeted towards charismatic species, the ones that capture our hearts and that we like to pay attention to, but is probably going to be ineffective to conserve ugly trees. And so next slide. So I've kind of set up a classic bait and switch. I've probably convinced most of you that coarse filter conservation is the right way to go. The problem with this is that ecosystems are transient, and I probably don't have to remind most of the physical scientists in this room that our environments change a lot through geologic time. And so this is where the Sundarbans are today, and you can see at the last glacial maximum, they would have been close to a couple hundred kilometers away from the ocean. And that would have, obviously the mangrove forest ecosystems wouldn't have been able to survive inland that far. And so I've kind of set up now what is termed conserving nature's stage. And so in this perspective, instead of conserving that ecosystem, you would try to protect all river deltas, for instance, or all places where rivers meet the coast, because these areas in general are highly productive and biodiverse ecosystems. So the idea here is that you conserve the physical stage by which ecosystems can occur, rather than focusing on single species or actors on the environmental stage. And that could even be applied to ecosystems. And the idea here is that abiotic diversity is more stable through time than biotic diversity. I'm going to use the terms abiotic and geodiversity kind of interchangeably. And so you can imagine these mountains here, they're not going to go anywhere even if climate was to change. And furthermore, having increased biodiversity can help increase and foster, or having increased geodiversity can help foster biodiversity over a range of climatic conditions. So you can imagine this is from the Olympic National Park. Species in this ecosystem, if it was to warm, they're going to be able to move up the mountain and they're still going to be present in that national park. And so that would be a good reserve design. And so various surrogates that you could, various geodiversity surrogates you could use for abiotic or biotic diversity would be climate, soils, bedrock, any sort of landforms or water bodies you could imagine. And so these would be the things that you might argue you ought to be looking at if you want to help conserve biodiversity. Next slide. And so there's actually been studies showing that there's a very strong correlation between geodiversity and biodiversity. This is an analysis of some northeastern states. And what they showed here was that the state with the most geologic bedrock classes, that's what the colors are in the map, also had the greatest biodiversity. And they got a scary good correlation, 0.94. This isn't of reality, this is of their model. So this is predicted number of species versus actual number of species. So a little deceiving there, but the point is their model was able to very well predict biodiversity just by knowing the geodiversity of that area. Next slide. And so I want to ask, we know that geodiversity is very important for biodiversity, but we don't know how well our protected areas are conserving geodiversity. We might like to think that our protected areas are some of our most geodiverse places, but we're also humans and we have biases. Much like we do with endangered species, we oftentimes pick the ones that are cute and cuddly or capture our hearts and minds. We might also be doing the same with geodiversity. We might be selecting types of geodiversity that we like. It may not be getting a representative pool of the geodiversity that's available. And so I want to look to see how well we capture that available abiotic diversity in our park systems. And moreover, to see if there's types of abiotic diversity that are underprotected and aren't available in any reserves. And then furthermore, see if abiotic and biotic diversity has that strong correlation within our national parks. So this is the data set that I'm planning on working with. It's an incredibly comprehensive data set on all of our United States protected areas. And so you can see there's all sorts of different types of protected areas in this data set. And it has just about all of them as far as I can tell. You can go next. And again, we're going to zoom into Maine. And you can see here all the protected areas that we have in Maine. And we have a kind of diverse set. And if you go in again, just because I know you'll be interested, one more. Here is it for Orono. And so I want to give you just kind of an impression of how dense this data set is. It goes all the way from our country's largest national parks all the way down to Orono City Forest. And so it's going to be that's going to provide a challenge to analyzing this data set. Next, please. So I'm planning on using many different geodiversity classes in order to kind of get our sample. I'm going to use bioclimatic variables. So these are climate variables that are thought to be important for ecology. These are things like your annual precipitation, your annual mean temperature. I'm also going to be looking at the Köppen-Treuertha climate classifications. These are climate classifications based on local vegetation types. As well as I'm going to also look at soil types and lithology and bedrock to see if those have a correlation or just to see how well our parks are protecting them. And this is where I turn it over to you guys. I would like to know if you know of any very good data sets on an aspect of geology or something like that that you think might be important for ecology. Please tell me, because I'd be interested in maybe including it in my analysis. And so I saved the most complicated slide for last. How am I going to do this? This is obviously going to be a very multi-dimensional, lots of different data. And so I'm going to be using an approach called a hypervolume approach. So hypervolume is, I can't picture it for you, but I have a nice spinning cube to picture a three-dimensional volume. A hypervolume is a region in more than 3 dimensions. And this is central to the concept in ecology of a species niche. It's n-dimensional hypervolume. And so what I've shown over here is these are plots of traits in Darwin's finches. And so these are things like tarsus length, wing length, beak length, and they have 9 different trait measurements. It's actually kind of impossible to see how well those overlap. You can look at 2 at a time, but that's not looking at it in multi-dimensional space. And our geometry is limited to kind of 3 dimensions. It kind of breaks down when we get to high dimensional space. It's just very difficult. And so there's actually been some recent developments with this R package, which allows you to do that. And so what they've done here is they've looked at these multidimensional spaces, and they wanted to test to see how well the niches overlap. You know, Darwin originally made the prediction that these finches ought to be diversifying in such a way that they each occupy a different, distinct niche, and that every one of these will be unique flowers. And what they actually found with this approach was that Darwin was mostly right. All the species pairs that you could look at, all of them are red, which means that they had 0 overlap, except for one species pair, which had a minimal overlap in between them. And you wouldn't have been able to pick that out, or at least I'd argue, with your own eyes, looking at all these different data sets to make sure that each color group, each color group is a species here, is non-overlapping. But when they ran it through the analysis, they found that that was the case. And so this hypervolume package now allows us to measure these multidimensional spaces to get a grasp of how big they are and also see if they overlap. And so you might imagine that I'll have climate, geology, and maybe another climate variable or soil variable on this, and I'll be looking at not only the abiotic diversity that's possible, but which ones are in the parks, and also to see if there's any discrepancies between the niches of endangered species, because I can create these for any species really you want, and see if there's any overlap in between, or any discrepancies really, mismatches in between our endangered species and the environmental spaces that are available in our protected areas. And so with that, I'll turn it over to questions, and thank you for listening.
Pretty impressive.
Thank you. No one? Yeah.
Okay, so you mentioned topography a bunch of times on some of the slides. You didn't always repeat that word. So I'm just kind of curious, do you have an idea yet how you're going to characterize topographic?
Yeah, and if you have any better ideas than the one that I've got, let me know. But I was planning on using a DEM and also to look at, well, so topography would be, there's a couple different measures of topography. There's not only the altitude, but then there's also the aspect, which is important for ecology. Is it a north-facing or south-facing slope? And so these would be kind of the lines I'm going along with topography, and just kind of how the slopes are oriented, as well as what sort of altitudinal coverage they have. I would suspect that our altitudinal coverage is going to be pretty good, because I think our protected areas like to protect mountains. But that's just a guess. Yeah.
Well, I was wondering if the R package has a way to deal with multicollinearity, because so many of those are going to be correlated with one another.
That is actually a really good question. I don't have an answer for that.
Yes. But you will.
Yep.
Really interesting.
Thank you. Anybody else? All right, well, I know I'm keeping us from beers, and so I'll turn it over to that. Thank you.
Okay, just before we move on, I think it's been a really good day so far. There's more to go. I have only one disappointment, and that is there have been almost no puns. So those are coming tomorrow. I think, Jeff, you did the best, but you missed a real opportunity. You could have noted your acknowledgments, how much in hock you are to the bison farmers.
I'll use it later.
There you go. All right, we're now going to move over to the poster session. So I'd like to ask all the poster children to stand by your posters for the next 35 minutes. We'll then go to dinner in the cash bar, and then back here at 6 for the keynote alumni lecture.